It's a pleasure and privilege to open this exciting meeting, and what I've taken as a charge is to provide a little survey by way of introduction about aging, multiple parameters across societal, statistical, demographics, on through some of the exciting basic science, as you just heard, an attempt to bridge the gap between the science that we are embarking upon and its eventual translation. So, some figures that I think are impressive to all of us, even though we already understand and accept the dramatic aging of populations as we're seeing them. Illustrated here are two curves that are showing the percentage of populations from 1950 through projected 2050. The top line is those under 5 years of age. The bottom is those 65 and older. This is worldwide. And you can see the really dramatic changes that have been happening so that there's a decreased proportion of young children and increasing proportion of older adults. And right about where we sit now, for the first time in the history of the species, clearly, there's going to be a cross of these numbers. And then the trends will continue. So it's in a population that is aging. This is tribute to longer survival, also demographics of birth rates, but one way very dramatically to see what's happening to our population. Now, in terms of life expectancy, some figures which I think are of interest historically. This looks from 1840 through 2000. This first portion we're looking at here, 1840 through 1920. And what it plots is the life expectancy of the female population, because women still outlive men historically, for whatever nation had the record in that year. So you can note from an age of about 45 in 1840, there was an increase to over 65. Over 80 years, an increase in life expectancy of about 20 years. So every year, every 4 years, life expectancy up by a year. Now, if you were sitting here, or we were sitting here in 1920, and asked whether we thought a trajectory like this could continue, I don't know how many of you would say yes, but in fact, if we plot it out through the near present, remarkably it has. And I don't know, maybe someone has a good explanation of why this should be so linear, because at each of these stages in history, the reasons for the increase in life expectancy are very different. Earlier, having to do with childhood mortality, later on, late life disease. But I just, as a thought exercise, there's no right answer, I think. For those who are going to be back another 40 or 80 years from now, whether this is going to continue or not. This is average, not maximal life expectancy. Clearly a lot of discussion about whether we're finally reaching a plateau at a maximal life expectancy, but quite dramatic findings over life course. Now, just some reality testing, and very sober reality. If we look at what has happened in this country, the United States, here illustrated from 1980 through the present, with the U.S. life expectancy compared to the comparable OECD numbers. And you can see back in the 1980s, they were very close. There's been an increase in life expectancy, but look at the gap. That is, the U.S. has done far less well than other countries. And if you break that down further, just to share a couple of the demographics, look what's happened as a function of socioeconomic groups. So this is men on the left, women on the right. We can look at women, for example. For the richest 10%, this is the increase in life expectancy from 1920 for birth year 1920 through 1950 cohorts. As you go through lower income, you can see that decrease is in turn decreasing, and in fact, for the poorest 10% of women, average life expectancy has decreased. For men, the picture is similarly bleak. So quite dramatic inequity of what has happened in terms of life expectancy, and with it, health expectancy as well as a function of socioeconomic levels. Now, just one more to this theme. Very well aware of the specific issue of so-called deaths of despair having to do with alcohol and drug abuse. What's shown here is in a study from Case and Deaton, with cohorts that were born in the year shown, so from 1935 and moving towards the left, more recent. For those cohorts, looking at what's happened to life expectancy and the proportion of deaths due to these issues of despair, that is drug abuse and alcohol. And you can see from to the right, the 1930s and 40s on to the present, ever increasing, dramatically increasing rates of death for these causes. But note, this is for those individuals who have less than a BA, that's a bachelor's degree of education. If you look at those with another education, and again, this is one of the most incredible contrasts that I've seen as a result of education. So education, all that comes with it, what its correlates are, extraordinarily important, but shows that there are undoubtedly malleable aspects of life expectancy, health expectancy, that we can work upon and deal with. So just some examples of translation, some places where we have learned there's some things that we can do. First, to look at the causes of death that rank first as of 2017. They're shown here, the top 5 you can see, the top 10, all common. We can pick a couple of examples of cases in which we've learned some things we can do to make progress. So we illustrate them here. You heard an introduction, the fact that there are not just drug interventions, but lifestyle interventions. This is an example of the study, the LIFE study, which is a rigorous, randomized controlled trial, which looked at the effect of an exercise program on maintaining absence of disability. In this case, this is mobility disability. So the end point was maintaining the ability to walk a distance over time. And the clinical trial, in fact, was successful to the degree where it would have been a drug. I think it would be widely applauded. It should be as an intervention here, too. So there's about an 18% decrease in mobility disability in the elderly as a result of this 2.6-year intervention. So there are clearly lifestyle interventions, and importantly, they can and need to be tested in rigorous fashion, as this is and as other ongoing studies are doing. Cognitive function, as important, I think, to many of us as physical functioning, illustrated here. This is an older study already, that is, was initiated some time ago, supported by NIH and NIA, the ACTIVE study. And what it did is, over a period of months, train people in particular aspects or domains of cognition, and those included memory, reasoning, and speed. And what you can see at the top is that training in reasoning had the effect sustained here, as shown over 10 years, of maintaining an advantage when subsequently tested. So there was a domain-specific and remarkably enduring effect of a few months of intervention. Down below, training for speed of processing and reasoning, an even more dramatic increase that sustained itself over time. And the most recent study of what happens 10 years later after this training, look at the impact on maintaining the ability to conduct activities of daily living. So the lowest figure here, the closed circles, are the control group, and over time they are decaying in their ability to maintain independence, activities of daily living. All those intervention groups with training in speed being the highest, able to sustain better cognitive function in that domain. And now the question is really whether this kind of intervention will help over longer term and to maintain not just normal abilities cognitively, but avoid the pathologies of Alzheimer's and related dementias. So in fact, a couple of years ago, at a time when there has been so much growing interest in identifying interventions, maybe public health campaigns, to use means that would diminish the risk of cognitive decline and or dementia, we undertook a study first carried out by Evidence-Based Practice Center through ARC, handing its results after it analyzed all the published literature over to a committee of the national academies, and asked whether there is a level of evidence for any intervention, again, decreasing rate of cognitive decline or dementia, that warranted a public health campaign. The outcome was that there was insufficient evidence to justify a public health campaign for any intervention that had been tested and reported to date. But there were encouraging, although inconclusive evidence in several areas. One was cognitive training, and the study I just showed you is an example of something promising. And so there are now studies being planned and ongoing to test this more rigorously. Blood pressure management and hypertension, and we'll talk in a minute about some results that came, in fact, after this National Academy's report, and increased physical activity. And here, too, a topic along with the life study I showed you, which is being sustained now with a large and increasing number of lifestyle interventions, single lifestyle interventions, or composite. So what about blood pressure control and hypertension? Now, some findings many of you may be well aware of came from the SPRINT trial. This was a trial that compared the effect of controlling systolic hypertension to a level of 140, which up until that time had been regarded as a reasonable and appropriate standard, or a more intensive therapy, reducing systolic pressure to 120. This was stopped prematurely because ethically it was determined it was impossible to continue the study due to the fact that the more intensive treatment for blood pressure had a substantial increase, 25% or so, in both life expectancy and in cardiovascular major events. At the same time the study was carrying out, we made sure that there was inclusion of older individuals and measures of cognitive testing as well as brain imaging to see if this intervention might have an impact there as well. And remember, the study was stopped early, therefore it wasn't the follow-up on treatment that was expected, but when the data were analyzed years after, despite the termination early, what was found is that although, and I have to stress, the primary declared outcome was dementia, there was a trend, but not significant decrease in the trend to dementia. But for mild cognitive impairment, which is a well-recognized, often precursor of dementia, there was a significant 19% decrease of new cases in the group with a more intensive blood pressure control. And if you looked at a combined outcome of dementia and MCI, again, a 15% decrease in those endpoints. And in fact, those patients who received MRI showed that the more intensive intervention decreased the accumulation of white matter densities, hyperdensities associated with brain lesions of cardiovascular origin in particular. So an example of the kind of progress that's been made, I think, even since that 2017 report. We'll continue to assess what we know, what we need to know, but here you can see both pharmacologic interventions for hypertension as well as lifestyle interventions showing substantial promise. A recent study, just point out, asked whether for individuals who had high or low genetic risk of dementia, whether lifestyle made a difference. And here a composite index for a cumulative risk score for dementia separated individuals into high, low, or high, intermediate, or low genetic risk. And then for each of those, looked at the impact, these data largely from the UK Biobank study, and found encouragingly that for all levels of genetic risk, even those at the highest genetic risk, there was an impact of lifestyle. Now, this is observational, needs to be translated, of course, into direct tests and interventions, but it points out the fact that for many of the adverse consequences that can come with aging, that genetics are not a pre-sentence, or they are predisposition, but subject to modification by lifestyle. So an encouraging bit of evidence. So now turning a bit to the geroscience concept we'll be hearing about this morning and throughout this session. There are the founders as well as advocates of geroscience here in the audience, and I hope they'll be participating in the study. But let me just summarize. The biology of disease, shown here to indicate a large number of diseases that surround them, diseases for which aging is a predominant risk factor. The biology of aging, including many biologic phenomena, some of which are illustrated here. The notion of geroscience is the conversion of these on the notion that it may well be that the high degree of risk factor of aging for multiple adverse consequences of aging is a consequence of the processes that occur quite broadly, if not universally, with aging, and that intervening with these basic underlying processes may not have a positive impact on just one disease, condition, or pathology, but on many. So I'd like to turn now in the remaining few minutes to give an example of just one of the targets that has been studied in years recent, and the really dramatic basic science and now the cusp of interventional testing, and that has to do with the analysis of senescent cells, so-called senescent cells, and senolysis, which is the lysing or killing of senescent cells. Now, what are senescent cells? Really, the term senescence, or here replicative senescence, came with the realization in 1961 from Hayflick and colleagues that cells, in this case in tissue culture, were not immortal. Now, up until this time, it was thought if you took normal cells, fibroblasts from the skin, commonly used, put them into tissue culture, they would grow forever. What Hayflick showed, and others have clearly reproduced, is that that's not the case, and that after a predictable number of cell divisions, cells stop dividing. And that state, largely irreversible, is called cellular senescence. Now, importantly, there are a number of factors which can lead to senescence of the cell, some of them illustrated here: telomere erosion, DNA damage, oxidative damage, loss of tumor suppressors. And senescent cells don't, importantly, just sit there passively, but rather importantly, they are active, and among the activities, well recognized now, is an altered secretome, that is a set of secreted products called the senescence-associated secretory phenotype. Many of these are pro-inflammatories and have consequences far beyond the scope of the individual senescent cell itself. So an illustration here of many of the secreted products that are increased in senescent cells in both mouse and human studies as illustrations. And then important studies over the last few years. This begun in the lab of Ned Sharpless, who was an investigator early in this work, supported by NCI and NIA, was on the advisory council of NIA till he had to give that up to become the director of NCI, and then left NCI to become now the acting commissioner at FDA. But this is some of the work that came from his lab, and what Ned did was take advantage of the fact that P16 is one of the genes that is increased consistently in cells that acquire this senescent, replicative senescent phenotype. And so he developed an assay, a reporter, in which cells that had high P16 could be measured. And on the left, you can see the studies his lab carried out looking at T cells from human peripheral blood, that the proportion of P16 expression, the intensity of P16 expression increased with age. He also had a reporter that could be put into a mouse as a transgene, such that the cells that were senescent, expressing high P16, glowed yellow, and you can see that here. And the plot in the bottom right shows this luciferase, or yellow cells, were increasing with age as well. So these are cells that have a senescence phenotype. They're increasing with age. Now, importantly, one of the aspects of senescent cell biology is not only do they have this secretory phenotype I mentioned, they're actually quite resistant to death. They don't die. And so targeting to see what might happen if one could alter these senescent cells in vivo was largely aimed at killing those cells which otherwise were resistant to death. And that was done first in some studies using a very clever genetic trick. A transgene was put into these mice, which was driven by P16, so it's turned on only in the cells that are expressing high P16. That has the equipment or machinery for killing the cell, but that machinery is activated only when a drug is given. So these animals live, they develop P16 expressing senescent cells, and at a point in time, a drug is given which leads to the activation of an enzyme called caspase 8, that kills the cells. So you can eliminate senescent cells quite acutely, however many have accumulated at a point in time. And I should note in some interesting human studies, estimates of the number of senescent cells increase with age, multiple tissues, but they're single digit. They're relatively infrequent cells, and yet eliminating them, we'll see, was tested to see if it might have an effect on the whole animal. And shown here is the effect of giving drug, treating these animals at a relatively older age, after a year of age, over 300 days. And at that point even, with that relatively late administration, so eliminating the senescent cells that had accumulated at that point in time, a significant extension that you can see in life expectancy, 25 and 18% in these experiments. So eliminating senescent cells, although they are in small proportions in many tissues, extends life. What else does it do? This is showing here first that in tissue-cultured cells, the staining for beta-galactosidase, which is one phenotypic measure of senescent cells, is eliminated with this same drug treatment and genetic trick. And on the upper right shows that in a tissue, when you take it out, that you've eliminated the beta-galactosidase staining cells. But here is one example of looking at specific tissue or functional assays. If you look at the gastrocnemius or abdominal muscles in two different lines of mice, what you see in each pair of bars there is that the fiber diameter of muscle is increased when you treat with drugs, eliminating that small percentage of senescent cells. And on the right, if you translate that into what happens to the ability of those mice to run over time, distance, and overall work, their functional capacity is increased as well. So eliminating the single-digit frequency of senescent cells has increased the capacity of these mice to function. One, I think, particular for an interest in metabolics and metabolism is illustrated here. On the left, a glucose tolerance test. What happens when you give glucose to an animal and then measure its blood glucose level in the mouse? So on the lower bar, chow, that's a normal diet. The upper bar is a diet-induced obesity. And diet-induced obesity is accompanied by abnormal glucose tolerance. If you do all that in a mouse that has this genetic machinery for deletion of senescent cells, you can see that treatment to delete senescent cells has the now diet-induced obesity mouse with a glucose tolerance similar to the non-obese mouse. And on the right, showing something similar, improving HbA1c, the same thing is shown, so an increase in HbA1c in the diet-induced obesity, which in this study was reported to be near completely reversed by eliminating senescent cells. A final example, because cognition and what goes on in the brain is so important. In this case, the treatment was targeted, once again, same genetic trick, at a neurodegenerative model in which there's a tau pathology that results in a decrease in the here shown dentate gyrus of the brain, an area very important for memory. And so you can see, again, in the bar graph, with pictures on the left to go with it, the size of the dentate gyrus area in white in a normal animal, and the animal that is transduced with a gene so that it now has a tau pathology, similar to what happens in Alzheimer's disease in the brain. The red shows the decrease in the dentate gyrus, and that same mouse in which senescent cells, here presumed to be not in neurons but in glial cells in the brain, in the study reported here, there's a restoration to the near normal level in dentate gyrus. And if you look what that means for function, in this case, a learning or discrimination task. So the animal that has the suicide gene has, as you can see, the animal that has the suicide gene has a low learning rate in white. So the training test is where you learn to do the task. Then in gray is how you do after you've learned the task. So going from low to high is learning in these two bars. In the mouse, the AP mouse that has the lesions of tau that resemble those of Alzheimer's disease, the white and gray bars are the same. The animal never learns to discriminate. But that same animal on the far right, in which senescent cells in glia are eliminated, now looks like a normal mouse. So preservation of the dentate gyrus and ability to sustain discriminatory learning are maintained by eliminating senescent cells that are occurring normally in the brain of this model of tauopathy and neurodegeneration. Finally, I want to point out just as an important corollary that in some cases, and one always has to ask this, are senescent cells all bad, or do we have to be wary of eliminating senescent cells everywhere? This particular slide shows that in wound healing, a wound healing model, that in fact eliminating senescent cells, and I'll just point to the top left there, shows that treatment with drug to eliminate senescent cells actually results in longer time taken to heal the wound. So remodeling in wound healing, probably during development in other areas too, may involve senescing of cells as a normal physiologic mechanism. So a lot of excitement about the kinds of things that might be done here, but also with cautions. Now, everything I've shown you so far is this clever genetic trick. How do you translate that into something that goes on in humans? Well, I mentioned to you that one of the characteristics of senescent cells is that they don't die. They're resistant to apoptosis. So what's been done is to screen, initial screens largely in vitro, take tissue culture cells that senesce and see what drugs will now make them die. And the combination shown here, dasatinib, which is an anti-cancer drug, a tyrosine kinase inhibitor, and quercetin, which is a supplement that is widely available and used too, a polyphenol. Two drugs, therefore, that are available in combination have proven to date to be one of the more effective eliminators of senescent cells in vitro, and hence these studies in vivo. So in this case, given biweekly, which is probably overkill given the pharmacokinetics of these drugs, show that again, if you begin treatment early in life or even late in life, out at 700 days in the life of a mouse, that combined treatment of these drugs extends lifespan, presumably through elimination of senescent cells, or that is, at this stage, senescence hypothesis. So we now have a circumstance in which the genetic manipulations and models indicate that eliminating of senescent cells has some very exciting consequences in disease models and overall survival. We have the beginnings of drugs being screened and widely screened to mimic the effect of that genetic trick and lyse senescent cells. And I'll just finish here with an update of where we are to date and emphasize how quickly this has all happened. These are early pilot studies looking in a variety of conditions at the effect of drug combinations targeting senescent cells for their effects. So far as you can see, some of them are given systemically, others locally, like in joints for osteoarthritis. The first measured but rigorous beginnings to identify the safety of these in humans and ultimately their consequences in individual pathologies and by extension perhaps the multiple exologies per the Geroscience hypothesis for processes that may underlie multiple diseases. And I'll finish just here with a note that for those interested in Geroscience and the progress that's being made and some of the excitement, there will be the third Geroscience Summit at NIH, November 4th and 5th, and invite any of you interested to come and participate. So thank you again for the opportunity, and happy to turn it over to my colleague.